Hey everyone! Welcome back to Pharma Show by Seji. Today's topic is quality control tests for ophthalmic solutions. What are ophthalmic solutions? Ophthalmic solutions are sterile liquid preparations designed for installation into the eye. Because the eye is such a sensitive organ, quality control here is extremely critical. Even minor contamination or formulation issues can lead to irritation or even serious damage. So, what tests are performed? Let's break it down. Key quality control tests. 1. Sterility test. What it is, confirms that the ophthalmic solution is free from any viable microorganisms, bacteria, fungi, or yeast. Why it's important, ophthalmic products bypass natural body defenses. Even a single microbe can cause severe eye infections or damage. How it's tested, performed under aseptic conditions as per pharmacopoeial standards. 2 common methods. Membrane filtration. Solution is passed through a 0.45 micrometers sterile filter. The filter is then incubated in fluid media to observe microbial growth. Direct inoculation. A small amount of the sample is added directly to culture media and incubated. 2. Clarity and particulate matter test. What it is, ensures the solution is clear, colorless, and free from visible particles or haze. Why it's important. Particles may cause mechanical irritation or damage the sensitive tissues of the eye. Ensures visual acceptability and uniformity. How it's tested. Visual inspection under a white and black background with adequate lighting. For subvisible particles, light obscuration or microscopic methods are used as per USP. 3. pH testing. What it is, measures the acidity or alkalinity of the ophthalmic solution. Why it's important, the natural pH of tears is around 7.4, a solution that's too acidic or basic can cause pain, tearing, or inflammation. How it's tested, performed using a calibrated pH meter. Buffer systems may be used to maintain desired pH within a narrow range, typically 6.5 to 8.5. 4. Osmolality test. What it is, checks the concentration of solute particles in the solution, expressed in mosm/kg. Why it's important. Human tears have an osmolality of 300 mosm/kg. A mismatch can cause ocular discomfort or damage to the corneal epithelium. How it's tested, measured using an osmometer. Example, freezing point depression method. Target osmolality is typically 270 to 330 mosm/kg. 5. Viscosity test. What it is, measures the thickness or flow resistance of the solution. Why it's important. Viscosity affects retention time on the eye and patient comfort. Too thin equals rapid drainage, too thick equals blurry vision or stickiness. How it's tested, tested using a viscometer. Example, Brookfield viscometer. Viscosity is often adjusted with agents like hydroxypropyl methylcellulose. 6. Assay, API content. What it is, confirms the correct amount of active pharmaceutical ingredient is present in the product. Why it's important. Too much equals toxicity risk, too little equals subtherapeutic dosing. How it's tested, typically done by high-performance liquid chromatography or UV spectroscopy, must be within the pharmacopoeial acceptable range. Often plus or minus 5 to 10% of labeled claim. 7. Preservative efficacy test. What it is, tests the effectiveness of preservatives in inhibiting microbial growth over time after the container is opened. Why it's important, multi-dose containers risk contamination. Preservatives like benzalkonium chloride must be proven effective against microbial threats. How it's tested, a known quantity of microorganisms, example, E. coli, S. aureus, P. aeruginosa, C. albicans, is added to the solution. Samples are taken at multiple time intervals, example, 6 hours, 14 days, 28 days. Reduction in viable counts as compared to pharmacopoeial standards. 8. Drop size and volume testing. What it is, this test measures the size of each drop dispensed and the number of drops per container to ensure consistent dosing. Why it's important, Patients typically administer 1 or 2 drops at a time, and each drop must contain an accurate dose of the active ingredient. Too large a drop can cause overflow and wastage, too small a drop may not deliver the therapeutic dose. How it's tested. The ophthalmic solution bottle is held at a standard angle and height. Drops are dispensed and collected on a pre-weighed surface or into a volumetric device. The average drop volume, in microliters, is calculated from multiple drops. Also, the total number of drops per bottle is counted to verify it matches the labeled content. Example, a 5 ml bottle should give about 100 drops if the average drop size is 50 microliter. 9. Container Closure Integrity, QCI, Testing. What it is. This test ensures that the container and its cap or seal are airtight and prevent any microbial or environmental contamination. Why it's important. Ophthalmic products are sterile, so any compromise in the container can lead to loss of sterility. QCI testing is mandatory for sterile products per global guidelines. How it's tested. Some commonly used methods. Vacuum decay test. The sealed container is placed in a vacuum chamber. Any pressure change indicates a leak. Dye ingress test. The container is immersed in dye under vacuum. If dye enters, the seal is compromised. Helium leak test. Uses helium gas and sensors to detect microleaks. High voltage leak detection. Detects liquid conductivity across a microleak using high voltage. 10. Compatibility with packaging. What it is. This test checks if the ophthalmic solution remains physically and chemically stable when stored in its intended container over time. Why it's important. The container material, plastic or glass, should not react with the solution. No leaching of chemicals, like plasticizers or stabilizers, into the product. The container must maintain the drug stability, potency, and sterility throughout its shelf life. How it's tested. Stability studies. Store the product in final packaging under various conditions. Example, 25 degrees Celsius per 60% RH and 40 degrees Celsius per 75% RH, 4 months. Physical compatibility. Check for color change, cloudiness, or precipitation. Chemical compatibility. Assay the drug content and look for degradation. Leachables/extractables testing. Analytical techniques like LCMS or GCMS are used to detect unwanted chemicals that may migrate from the container into the solution. Conclusion. Ophthalmic solutions are a perfect example of pharmaceutical precision. Each test from sterility to pH plays a role in protecting your eyes, and as QC professionals, we ensure that every bottle meets the highest standards before it ever reaches the patient. Thanks for watching. If you found this video helpful, don't forget to like, subscribe, and share it with your fellow pharmacy or QC friends.